Hello, this is Suman, and today's topic is Friedge fracture technique. It is a technique to study the plasma membrane, particularly to visualize what is inside the membrane. So let's begin. The name is Friedge fracture technique. So what is the meaning of Friedge? That means you have to make it frozen. Okay. So we know that the membrane is a fluid structure. The membrane is fluid. So to make it solidified, you need to freeze it. The first step is to solidify. In this technique, the cells are quickly frozen in liquid nitrogen, which immobilizes the cell component instantly. So the cells are frozen in liquid nitrogen. Then the second step is, then you need to cut it with the knife. So the frozen tissue is fractured by the help of a knife. So the second step is to cut the frozen tissue. Third point is Friedge etching. What is it? It means the surface ice is removed by the vacuum. By the help of vacuum, the surface ice is removed. Okay. Then the fourth step is a thin layer of carbon is evaporated vertically onto the surface to produce a carbon replica. So production of carbon replica. Next is surface is shadowed with a platinum vapor. The exposed membrane is then coated with the platinum. The next is the organic material is digested away by acid, leaving a replica, and in last the carbon metal replica is put on a grid and examined by a electron microscope. So the steps are, first you have to make it solidified by freezing, then you need to cut it by the help of the knife, that is the fracture. Then the fracturing cause one half the membrane to separate from the other. So what will happen when you fracture? You can see the separation occur. The membrane, one half of the membrane is separated from the other half. That is the splitting of the phospholipid bilayer into two separate layers. Okay. Then you need to remove the surface ice via the vacuum, that is Friedge etching. Then a thin layer of carbon is evaporated vertically onto the surface to produce a carbon replica. Then you need to coat the platinum to the exposed surface of the membrane, and at last you can visualize it by the help of a electron microscope. So the purpose of this Friedge fracture technique is to visualize the inside of membrane, particularly the ultrastructure of the membrane interior, the pattern of the integral membrane proteins. So this is all about the Friedge fracture technique. I hope it makes sense to you. Please do subscribe and share with your friends. Thank you.